Hi, I'm here with Dr. Nick Glover. Nick is a consultant to the biotechnology and life sciences industry with over 25 years of drug discovery and development experience. Nick, thanks so much for joining us at MaRS Impact Health 2022.
Oh, it's my pleasure.
We're gonna dive in, and the reason you're here and the reason I'm really excited to talk to you today is about Sierra Oncology and its recent success, and we're going to dive into that in a minute. But for the audience who doesn't know you, because I know you before the show you said you've been around for a while, maybe just give us a thumbnail sketch of your background and training.
Yeah, absolutely, I'd be happy to. So my training is actually in medicinal chemistry. I've been involved in drug discovery my entire career, starting with my academic career, which was an industrially oriented PhD thesis, working with Merck Frost in Montreal when there was a Merck Frost in Montreal, and continuing through my initial foray. I broke into the industry actually in venture capital, working with a venture capital firm in Toronto that was known at the time as MDS Capital. It's now known as Lumira Ventures. And then moved out of venture capital into the operating side of the business. I was very keen on being involved in hands-on operating drug discovery and drug development, and have been involved in that field ever since. I've had the pleasure of running multiple public companies, sitting on public company boards. I've been a public company CEO for about 17 years now, and continue to work very much at the forefront of leading biotech entities, always small companies. I've never really been interested in working for a large pharmaceutical company. And I think that's apropos to this storyline, to be fair. And I continue to be actively engaged in the industry. So very excited about the current state of affairs in the Canadian life sciences industry, and very much keen to continue to contribute in any way, shape or form that I can.
It's fantastic. There's a lot we could dig in there. But for today's purposes, and for those in the audience who don't know, we're going to start with the punchline, which is Sierra Oncology, which was, I think, the last venture that I'm aware of that you were associated with, was just acquired by Glaxo this year for $1.9 billion. And the journey, as it never is in biotech, was never a straight line, but this passed through a lot of hands. And I think there's a lot of interesting learnings potentially in there. And to me, that's the fascinating part of this story and why we wanted to have you. And the journey, correct me if I'm wrong, Nick, started really with one asset, momelotinib, originally positioned as a JAK1 or JAK2 inhibitor for myelofibrosis. It started out as CYT387. And here's where I think it's really fascinating, is it changed multiple hands from Cytopian in roughly 2008 to YM Biosciences in 2009, to Gilead in 2012, to Sierra Oncology in 2018, and then finally its most recent acquisition by GSK in 2022. I believe you were part of at least two of those firms, correct me if I'm wrong, YM Biosciences and Sierra Oncology. So my first question is, maybe give us a brief background of Sierra Oncology first, and then tell us, why were you so persistent on this one asset?
Yeah, no, absolutely. I mean, obviously, I had the pleasure, just taking one step back at YM Biosciences, of picking up what was at the time, as you said, CYT387, and being part of that initial translation into the clinic at a point in time in which JAK inhibitors were very much, an early stage and unproven science, but certainly a very promising one. And now JAK inhibitors clearly are bona fide agents across a variety of indications. Cytopia, which was the company that we acquired at YM Biosciences, was actually a pioneer in the JAK inhibitor space. It was founded by Dr. Andrew Wilkes, who was the discoverer of the JAK kinases. Okay. So in many ways, it was the prototype JAK inhibitor company developing the prototype JAK inhibitors. So it had a storied past and obviously very deep science with respect to the JAK space. Translating the drug into the clinic, it became very apparent early on that the agent was differentiated versus other JAK inhibitors that were further ahead in clinical development. And I think that's a really important part to stress. And certainly to me, one of the most important things for entrepreneurs in life sciences to be keenly aware of, which is differentiation. How is my asset different from other assets? And answer that question in a very frank way. In our case, it became very apparent that our drug did something that the other drugs in the space didn't do, which was to have a positive effect on anemia. Myelofibrosis, as the name suggests, is a disease of bone marrow fibrosis. These patients have impaired bone marrow, and impaired blood production as a consequence of that. And in fact, most of the JAK inhibitors actually exacerbate this. They make it worse.
Okay.
Our drug didn't. Our drug did the opposite. So it became very apparent very early that we did something different. Now, at the time, to be fair, we didn't quite understand why. We just knew that we did empirically. And so the question of why persist with it, I think fundamentally comes down to two things. One, it was clear that this was a differentiated asset. It was doing something different biologically. And secondly, at the point of time of reacquisition, I sold the company to Gilead via Biosciences, and obviously continued to enjoy good relations with Gilead. When it became clear that Gilead was no longer interested in developing the asset, of course, the opportunity to bring it back was incredibly attractive. I was fortunate enough to convince my board of directors that we should do that at Sierra. We brought the asset back in, and obviously the rest is history in teeing it up for success. And obviously now it's going to be in the hands of GSK moving forward. I think that's the right thing for that agent at this point in time. So differentiation. But secondly, at the point of reacquisition, it was very clear that this was a drug. Now, in my parlance, what is a drug? A drug is an agent that's proven itself, and by proven itself, I mean it's been demonstrated to have the following three facets. One, demonstrable efficacy, frank efficacy. It's an agent that clearly does something in terms of the indication that we're looking to treat. Secondly, that it has durability. So it's one thing to have an immediate effect, it's something else to have a long-lasting effect. And hand in glove with durability is tolerability. And this is an agent that's been proven not only to have an immediate effect, but have a long-term effect that can be sustained not just for months, but for years. So when we brought the agent back in, it had been in some patients from the initial clinical translation. Some of those patients had been on drug therapy 6, 7, 8, 9 years. So you knew that the drug was safe, it was active, and it was durable. That's the hallmarks of a drug. And it was differentiated. So it was really a no-brainer in terms of bringing it back in. So we knew what it had, we knew what to do with the agent, and obviously that's what we set out to achieve.
Perfect. So that kind of begs the question, I guess. So Gilead took it over, did 2 trials, Simplify 1, Simplify 2, mixed results, didn't hit all the primary, secondary endpoints. What didn't they see then that you saw? I mean, was it just the way they were positioning in it? Was the team what was the magic, I guess, if you will, that Sierra kind of said, aha.
Look, I think there's a couple of interesting lessons there, and certainly these are my opinions. You know, let me be clear. I think Gilead is a top quality organization, and Gilead did a lot of really good things with that asset, no doubt about it. So let's be clear, not disparage Gilead in any way, shape, or form. On the other hand, though, I think there are a couple of things here. You know, primarily Gilead's orientation as a behemoth in the industry.
Yeah.
And remember, at that point in time, Gilead had an incredibly successful hepatitis franchise and wanted to diversify into oncology. But their views on diversification into oncology was very much a go big or go home, because they needed to see, okay, what's going to be the successor to a multi-billion-dollar franchise that essentially was reaching saturation. And so their orientation was very much, I need multi-billion-dollar drugs that have the potential to replace revenue that we're going to be losing over the forthcoming years. And that very much tempered the way they designed the studies. Their studies were designed very much in a go big, go home way, which as a small co, it was not the way I would have developed the agent. It's not the way, frankly, that we'd set up the drug for continued development at YM. We had regulatory buy-in from FDA in EMA as to how to bring the drug to market. It was, in my view, a high probability of success approach in which essentially the drug was guaranteed of winning. The Gilead approach in clinical trial design was quite different from that. And so they took risks that I wouldn't have taken in the interests of commercial outcomes that, you know, in retrospect, miss some of the nuances in the agent. And I think that's the second part of this. Big companies have great people, no doubt about it, but those great people are spread thinly across generally portfolios of assets. They also have bureaucracy and administrative responsibilities that are quite different than you have in small co. In small co, we live and breathe our assets 24/7. you might have one or two assets. Generally, you only have a couple of assets in a portfolio if you're a small company, because you only have the capital to pursue a couple of assets. In our case, that was the most important drug we had. And so the entire brain trust of the organization was very, very keenly focused, not just on the agent, but on the indication and understanding the patient population, understanding what that population needed, and thinking about the nuances of that population in a way I think that really allows for sophisticated drug design in terms of the clinical trial strategy. That's what we brought to the table. Gilead's orientation was a big commercial company. Our orientation was very much a biology drug development organization that was very keenly focused on ensuring that the perturbation of the biology that our agent could affect would be translated into clinical trial success. And I think that's really the nuance here in the storyline. I don't think Gilead missed it. I think Gilead's orientation was quite different. And look, at the end of the day, they were incredibly gracious. They were very, 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 very supportive of us taking the asset back and finishing the job, because they believed, as we did, that there was clearly something here, and they acknowledged that perhaps their orientation had perhaps missed some of these subtleties that we very clearly knew and identified in the clinical trial designs that Gilead had prosecuted. The things that it had missed on, to me, had glaringly obvious flaws that were readily addressed. And that readily addressed strategy was translated into the momentum design, which was set up for success and obviously was successful. So look, I think there's a lot of lessons there. Taking a differentiated asset, understanding the biology, and really looking to ensure that clinical trial design was a very faithful representation of the clinical need, the biology that was driving that clinical need, and frankly, stacking the deck in your favor. And I think those are the lessons that you can take from that experience.
Fantastic. I'm going to ask you to look in your crystal ball for a second. You've been in the industry a while. How often do you think that happens, where assets are actually positioned in clinical trials, let's just call it, for lack of a better word, inappropriately, and kind of fall down on the cutting floor, and no one like Sierra Oncology has the foresight to go back and pick it up and actually do this again? What's your sense?
I think it happens all the time.
Okay.
I think it happens all the time, and I think the drivers are several fold. From a big company, the drivers are quite often commercial, and it tempers the view on how a drug should be developed and how what it should be compared against, and what the trial design should be in terms of driving a medical affairs effort, and so on and so forth. And all those things aren't, look, it's a commercial business. Those considerations are important, but perhaps they need to be balanced against fundamental biology and translation of that biology. I think for small companies, the opposite is necessarily the case, in which a small company, capital constrained, is quite often in such a rush to move things into the clinic, and believes that the translation into the clinic and the development of clinical data is so important that they miss some of the nuances on how that should be successfully achieved. And look, I think both of those factors get in the way and can obfuscate good biology. Poor trial design can absolutely obscure good biology, and it happens all the time. But at the end of the day, if you can match, and I guess this is my approach to drug development in general, if you can match the needs of the indication space, the needs of the patient that you're trying to treat, and understand how the biology that you're interested in perturbing has led to that clinical manifestation, then the opportunity to design a clinical study in a way that sort of faithfully represents both of those phenomena is sort of much improved. And then layer on the other considerations on top of that. But I'm very much a patient presentation and biology driven drug developer, and frankly, that's been successful for me.
Fantastic. So you mentioned, you like being in small cos. A lot of the audience is in small cos. They may have assets or starting programs, looking at sort of mapping out their clinical trials roadmap. Any particular advice that you would give for them to sort of navigate that a bit more efficiently? Or maybe you think it's done well now. What are your thoughts on that, having both the success as well as lots of experience in that area?
Yeah, no, absolutely. And look, I mean, I've had successes, but equally, I've had many agents that underperformed. And look, I think what's really important, in my view, is to try and mitigate the operational and strategic risks in drug development and leave the risk in the biology. Obviously, that sounds a little trite, but that's certainly what you're trying to achieve. And the way to do that, I think, is the formula that I've just laid out, which is to really understand the clinical indication that you're interested in treating, understand how those patients are presenting, and what kind of clinical benefit you'll need to achieve to demonstrate, not just to yourself, but more importantly to the regulatory bodies, that you have clearly made a difference in that patient population, and you've done it with an agent that is appropriately well tolerated, so that you've got the right risk reward balance. You've got a benefit risk in your favor, but you've really got to understand the patient population. And so take oncology. Does the patient population at the point in time that I'm intervening present with a single mass, with multiple masses? Do they have visceral disease, metastatic disease? Do they have ascites? What have I got to do to improve that patient's life in a meaningful way? And what has been the biologic basis for that presentation? And be very clear on understanding the biological link between how the patient presents and their underlying biology and what your agent does. And ensure that the way that your agent perturbs that biology has the potential to make a difference here. Don't fool yourselves with preclinical modeling that you're going to be able to make a difference because you can regress a tumor in an animal, or you have a particularly potent small molecule inhibitor. It's a lot more sophisticated and complicated than that. So I very much implore people to think about the clinical situation and work their way backwards. And don't fool yourselves into thinking that your preclinical modeling has the potential to make a difference here. Really stress the agent and ensure that you're in a position to stand behind a development strategy that's going to be able to move you towards potential regulatory approval. Again, it's not about rushing into the clinic. It's about working back from that clinical situation to where you are today and iteratively moving yourself forward in the development strategy. I think other elements of that I've stressed before is to honestly ask yourself the question, how am I differentiated versus other approaches? Like if you're the first thing out there, then that's an easy question. But you still have to compare yourself to what standard of care and what's emerging as standard of care, even if it's not directly in your drug class. But the notion of how am I differentiated is an important one, not just to yourself, but to your sources of capital, to the regulatory agencies, frankly, to every constituency that's important. The investigators that you're trying to convince to use your agent in the clinic, collaborators, potential partners, and so on and so forth. So the question of how am I differentiated and how do I reinforce my differentiation with as much data as possible is a really important one for entrepreneurs to have a keen eye on, in my opinion. It's something that I've always been very, very focused on in all of my development programs. And if you come out of that assessment going, I'm not particularly differentiated, then you have to take a long look in the mirror and say, do I have a path here? And that's the important thing to always remember. You can't be emotionally attached to your assets. If the data do not support that you have a path, they do not support that you have a path. And you have to be willing to say, it is what it is. That is the business. It's a business in which very few assets get to the finish line and most assets fall along the way. What we can do as drug developers in small companies is take a very rational and pragmatic view on what are the key questions that need to be answered at each step of the way, to convince myself at each step of the way that I still have the potential to get to a point in time with a differentiated asset that affects an indication in a meaningful way and has the potential not only to be efficacious, but to be durable and to have long-term tolerability. That's the momelotinib story. And that's the story that I think, many of us could take against other assets that are much, much earlier in development, but think it through to that point in time and how are we going to get there.
That is all fantastic advice. Thanks so much, Nick, for joining us today. And I look forward to seeing what comes next from Dr. Nick Glover.
Thanks very much.
All right.
Appreciate it.
Cheers.
Good luck, everybody. Bye-bye.
Okay, bye.